Hello, this is Josh at Dream Body Clinic, and today we're going to talk a bit about how we produce Muse cells in the lab. So Muse cells are found within mesenchymal stem cells. So we have to do the same process of producing the mesenchymal stem cells, which I'll get into here in a minute. But then we have to stress those cells because that's what Muse stands for. They're multi-lineage stressed enduring stem cells. They basically survive through stress that would kill mesenchymal stem cells. So that's part of the process. Now this goes back to how Dr. Tasawa discovered them in Japan. Apparently they were cultivating mesenchymal stem cells, which the first step is you take the umbilical cord, placenta, whatever tissue you're using, and you start dissecting that. You're then putting it into these cylinders with collagenase and centrifuging, spinning them real fast to separate the cells from the tissue. And after that, you would then normally take them and then centrifuge further to what's called the pellet, then put it in the flask, and then start culturing. Well, they just got to the point where they had centrifuged and something happened. They had to leave the lab and they left, forgot about them, went back the next day. And at this flask with all the collagenase and cells, well, as expected, the cells had died, fallen to the bottom, but there were still some cells that had survived that were at the top. So they're like, oh, wow, that's weird. These should be dead. Let's take a look. What are these? They analyze them with the flow cytometer and they start noticing these have the surface markers, phenotypes of pluripotent stem cells. Right? That's interesting. Research further. Sure enough, they're pluripotent stem cells, which means they can turn into any type of cell in the human body needed. So very interesting. The cool part is they act like mesenchymal stem cells, that they seek out inflammation and damage. They go there, they basically consume or take over the dead cells or injured cells and then turn into that tissue type. So really cool. But to make them, we have to first do the process with mesenchymal stem cells. Dissect, centrifuge down to the pellet, put it in the flask, and then we add culturing medium, which is like the food for the stem cells. We put them in the incubators, which are like, think of it like a specialized oven where you can control the temperature, the CO2 levels, oxygen, all that. And they start replicating, but they're eating through that culturing medium in that time. So the first passage, we do that. The MSCs adhere to the plastic of the flask, but there's a lot of other cells still in there. It's about 70% mesenchymal stem cells, 30% other types. So then once they eat through the culturing medium, we have to do what's called an enzyme flush. We flush out all those other cells. Luckily, the MSCs adhere to the plastic, so they stick around. And then we add more culturing medium. We go to passage 2. So once we do that, it's a passage. Now we're at passage 2, same process. And by passage 3, we've filtered out all those other types of cells. We're left with pure mesenchymal stem cells. Now this is the point that's different. Normally with mesenchymal stem cells, we'd go 1 or 2 more passages because we can keep replicating those MSCs. Well, the Muse cells don't replicate. We only get so many. We have to go to this third passage because we have to get rid of all those other cells. And now we start stressing them. So we've got vibration tables, we've got different ways to turn down the oxygen levels and stress the MSCs until they die, but take them just to the stress point where the Muse cells can survive. So it's a fine balance and it makes it really difficult because you can mess up a culture very easily, a Muse cell production, and you only get so many. It's not like MSCs where we can just keep replicating them, get more, do more batches. So we have a lot more work involved. This obviously brings the cost up. It also means we're throwing away or destroying a lot of mesenchymal stem cells, which is unfortunate just to get to these Muse cells. But we have to do that because what we found is there's a specific ratio at which the Muse cells act like Muse cells versus like MSCs. If there are 70% Muse cells to other types of cells or more, they will act like Muse cells. We're lucky we're getting like in the high 90 percentile, the cultures of purity for the Muse cells. So in that kind of scenario where it's almost all exclusively Muse cells, they act just like Muse cells. But if we were only getting 50% Muse cells, 50% MSCs, well, they would just act like MSCs. So we can't mix the two. So this is what makes it really interesting, a bit more complicated, a bit more costly for what you get, but there are advantages. First, they're a true pluripotent stem cell. And second is what's called the engraftment So with this, mesenchymal stem cells, when we put them into you, let's say we do a 300 million IV, we're hoping to fix your kidneys, let's say renal failure. I'm saying next, we had 2 patients for renal failure to do today with the Muse cells, actually. And so with renal failure, you're going to do an IV, it's going to go through your system, and we're going to hope we get enough to your kidneys to help them start regenerating. So with the MSCs, you have a few problems. First, they get stuck at the lungs for 2 to 3 hours. That's not good. A lot of them stay there, so you don't get as many through. And then you've got to hope the rest get to the kidneys and stay there. The engraftment rate for MSCs is around 1%. So that 300 million, maybe 3 million or so are only going to get to the kidneys, and maybe fewer if a lot gets stuck in the lungs or have other areas they're going to start going to. It takes 2 weeks to 2 months for the MSCs to decide where that engraftment area is. So it takes longer, the healing time takes longer. We usually see peak results 3 to 6 months after. With the Muse cells, a bit different. They're small enough they don't get stuck in the lungs, and their engraftment rate is much higher, closer to 15%. So though we're only doing, say, 20 million or 40 million, like for the patients today that did it for kidney failure, it was 40 million Muse cells, that's going to give them an engraftment rate of closer to, I think it works out to about 5 million Muse cells to that area. And the big advantage is they're going to differentiate faster, they're going to engraft faster, and well, not in differentiate faster, they do differentiate. MSCs don't differentiate. So these will actually turn into the kind of cells that you want, and they're going to get there, start eating and kind of taking over the dead, damaged kidney cells, and then start turning into kidney cells to replenish and replace that tissue. You get new kidneys, basically. So we're still at early stages of this. You know, we've had, I think, about 4 or 5 patients do it for kidney failure now, so we'll have some good data on that soon. We're expecting to see results faster and more of like the 1 to 3 month kind of time frame. So here in the next few months, I'll have some more data for you to really report back on that. But this is why it's more complicated to do Muse cells. It's how they work out. This is how we're able to do it. And it's more difficult. This is also why if you want to do Muse cells, we need a bit more heads up. With MSCs, we're every day got batches coming out. We're producing a lot, making sure we have enough every day. Typically have excess so that we can help last minute patients, things like that. With Muse cells, we need at least 2 weeks heads up, if not further, to make sure that we can start the process and then have the Muse cells available for when you get here. So if you want to learn more, check out the website at www.dbcmusecells.com, or you can call us toll free at 888-704-3977. I'm Josh, happy to help.